So without further ado, I'll invite Harrison Specht to come and tell you about some of the thoughts that we have in designing our experiments and some of the ways that we have found useful to analyze the data and validate them to make sure that what we get is not just noise.
Cool. So hi, everyone. My name is Harrison. I'm a grad student in the Slavov lab. And welcome to the first workshop of this afternoon. I'm excited to tell you guys about our efforts coming up about designing single-cell proteomics experiments with Scope 2. So before I get into Scope 2 and what that is, let's talk about single-cell omics type experiments in general. So in general, when you want to think about designing these types of experiments, you want to think about how many cells do you want to look at, how many genes do you want to profile, in our case proteins. And then after you figured that out, what sort of power calculations do you need to have in order to have significant statistics to make good conclusions? So a lot of this has been discussed thoroughly in the realm of single-cell RNA-seq, and I'm not going to go too much into that this afternoon. So rather, I'm going to delve into some of the unique quirks of doing single-cell proteomic experiments, especially with Scope 2. So what is Scope 2? Scope 2 is hot off the presses. Less than 24 hours, we posted it on bioRxiv, and it is our second generation iteration on the Scope MS approach to doing high-throughput single-cell proteomics. So the goal of single-cell proteomics is to profile many cells, as many as we need to, like we talked about before, while at the same time quantifying as many proteins as possible. One day, hopefully, the whole proteomes of single cells. So in a step in that direction, a couple years ago, Budnik and al. Developed Scope MS. And essentially, a lot of the core ideas that I'm going to describe as attributed to Scope 2 are ideas that were developed and implemented in Scope MS. What differentiates Scope 2, this sort of latest generation of Scope MS, is that we've made an effort to make Scope MS more high throughput and cost effective. Cool. So some of the core concepts, and I'm going to go through this step by step, and please feel free to stop me at any point and ask a question if any of it is unclear, because a lot of what I'm going to talk about later for the rest of the presentation builds on understanding exactly how this method works. So the Scope 2 method, and like I said, a lot of the ideas are exactly the same as Scope MS, tackles the challenge of delivering single-cell material to mass spectrometers. Now, mass spectrometers have been used very successfully for doing bulk proteomic measurements, measuring whole proteomes, and quantifying all the proteins in the proteomes. But the challenge for doing small samples and single-cell samples in the past has been delivering enough material to the mass spec in order to quantify the proteins in those small samples. So mass specs are incredibly or exquisitely sensitive. They can quantify as few as a handful of ions. So really, we have to deliver the proteins in our small samples to the mass spec. That's the challenge. So Scope MS addressed that challenge by using what's called a carrier channel. Now, the carrier channel reduced losses seen by our small samples by disproportionately absorbing them. So I'm going to take you step by step through the process to illustrate that concept. So first, we have our small samples, single cells or like, and we also have our carrier channel where we have a similar material, proteome, and we break open those cells. We digest them to peptides because we have a very strong ability to quantify and then identify proteins by quantifying and identify peptides. And then we reach the sort of crux of the methods. We barcode these peptides using a chemically identical barcode, but isotopically different. So now all of our samples are chemically identical to one another, and we mix them together. Now, by mixing them together, and since they're chemically identical, all the losses that are experienced by our peptides from our small cells, from our small samples or single cells, are going to be experienced in exactly the same way as the carrier. And the carrier, because it's so much larger, is going to absorb a lot of these losses. A lot of these losses happen along the sample prep pathway. Many of them happen in the process of doing chromatography. Chromatography is where we take our complex mixture of proteins or peptides and we simplify it by separating out our peptides in time and serially analyzing them with the mass spec. So then, once we have basically started to serially analyze our ions, our peptide ions, we can then identify them individually, isolate and fragment them, and then we sort of reach sort of a second key concept in Scope MS and Scope 2. And that's that the carrier channel is going to provide us with enough peptide fragments such that we can sequence the peptide. Basically, use the fragments as a fingerprint and map it back to the peptide sequence that it came from, and then we can map back that peptide sequence to a protein that it came from. Now that we have identified these peptides with the help of the carrier channel, we can also talk about the quantitation that we're able to do. Now, these barcodes I told you were chemically identical to one another, but they're isotopically different. So that means that they appear the same when the ions are whole, the peptides are whole, but once they're fragmented, they break into distinguishable fragments that we can then use to obtain relative quantitation from the peptide coming from each of the samples. So then, once we've acquired this raw data from our mass spec, we analyze it with MaxQuant. And part of the process of developing Scope 2 was to use this software platform that you're going to hear about later, developed by Gray Huffman, Albert, myself, and Nikolai, to iteratively optimize the sample prep and LC-MS parameters such that Scope 2 is delivering an optimal sample prep and method analysis platform. We also then take our data, and this is really cool. So you're going to hear about this on Tuesday. We're going to look at use some extra information and leverage the retention time of our peptides as they elute from our chromatography, and use that information to increase our confidence of identification, and therefore leverage and get deeper, more sensitive data. Shoot.
What about the carrier cells? Are they similar across the different samples, or how do you select?
So that's my next slide. Yeah. Cool. So I'm going to go into the carrier and address that question in 1 second. So the carrier channel, the idea here is that we're going to have enough material to reduce losses and aid identification. Now, something I haven't talked about yet is this reference channel that you see on here. Now, the reference channel is going to provide us with a consistent denominator, because I told you we're doing relative quantitation. And the barcodes that we use are commercially available TMT11 barcodes, of which there are 11. Now, that means if we want to analyze a lot of single cells, we have to create more than one scope set. And what I mean by scope set is a carrier reference and as many single cells as we have barcodes for. But the reference channel is going to provide us with a consistent denominator for doing the relative quantitation when we go to combine these sets that we've acquired at separate times. So how should we make our carrier channel? So here I'm showing 11 different proteomes from 11 different tissues of the human body, and basically correlating the quantified proteins that we can see in these different tissues. And we can see that they correlate pretty well, indicating that over the dynamic range of the proteomes in these tissues, they're pretty similar to one another. So that means that our selection of the carrier channel can be informed by, we want to select basically cell populations that are similar to the single cells that we want to analyze. And based off of this, many of the different cell populations in the human body would serve as adequate carrier channels for single cells coming from different tissues. So what we want for the carrier channel is for it to be representative of the single cell or small cell sample populations that we want to analyze. Why is that? It's because a lot of the identification that we're able to do using SCOPE-MS comes from identifying the carrier channel and then doing the quantification using the barcodes that come from the single cells. We're not getting identification from the single cells. So therefore, we have to be looking at an intersect of two proteomes. So we want that amount to be about 100 to 200 cells, enough to get good identification, but not enough to outcompete the ions coming from our single cells. This, like I said, can be prepared in a single batch or multiple batches because we're using it for identification. And batch effects are less likely to affect identification rates than they are to affect quantitation itself. And then a sort of unique feature that we can also add to the carrier design is that we don't necessarily have to come up with a cell type that has our proteins of interest and use that as our carrier. We can also use synthetic peptides or proteins and trigger measurement based off of those by spiking them in large amounts to our carrier channel. So a little bit more about the reference design. So this should be very similar to the cell populations you wish to quantify. It should contain approximately 5 cells, or a close enough number to the single cells that we have confidence that we were within a linear range of quantification when comparing the barcodes from the reference channel to the barcodes coming from our small samples or single cells. This should, unlike the carrier, should definitely be prepared in a single batch. And because we want to use it as a consistent denominator across all of the different sets that we want to quantify. And this also gives us the potential for estimating protein copy number per single cell. We can take a well-quantified synthetic peptide, we can spike it into our reference channel, and then we can use the relative amounts that we see in our single cells to extrapolate out protein copy numbers per single cell. So what would happen, sort of just to motivate the reference channel a little bit more, what would happen if we didn't use a reference channel? Let's say we go and we do a large experiment. We have hundreds of scope 2 sets, and we then had disparate cell populations. We randomly sorted them into our scope sets. We could then think about using, as a consistent denominator for relative quantitation, the mean of the single cells in each set. That sort of seems like it'd be similar to the reference channel. But then we didn't really control for the single cells that are in each set. It might be the case that we sorted cells of one type into a set and then sorted cells of a different type into a different set, in which case we'll have different means and different denominators, and therefore we can't combine those sets and compare them. We can also consider using the carrier channel, but we don't want to have to assume that we're within the same linear range of quantitation because we've used such a larger amount of material there. Cool. So those are the basic concepts of the Scope 2 and SCOPE-MS protocols. And now we're sort of going to go into where it all goes wrong. So I think anyone who's done experiments in the lab is familiar with this idea, whether they know it or not by name. And when we want to go and design Scope 2 or SCOPE-MS experiments, we want to assume that anything that can go wrong will go wrong. And sort of as a cool corollary to that, whatever goes wrong is going to be the worst thing possible for us. We want to go into designing these experiments with these two assumptions. Cool. So the first thing that we want to think about is designing our experiments in such a way that we can verify that we have succeeded in preparing good data and that we can trust it. And then the second thing we want to think about in designing experiments is how to diagnose where we failed. If we figured out we didn't succeed, how do we figure out where we failed and fix it? So it's these two principles that I'm going to talk about for the rest of the talk and start to address how we can do those with Scope 2. So the first way that we can verify if we succeeded is we can do external comparisons to measurements that we have more confidence in. So we can look at quantification in fluorescent proteins and directly compare measurements in single cells to measurements of the fluorescent proteins in single cells and measurements of the proteins by Scope 2 in the single cells. And then we can also, because taking these sort of measurements in single cells is difficult, we can also compare our measurements in single cells to bulk proteomics measurements, which we have much more confidence in our quantitative abilities to accomplish. So for example, in a SCOPE-MS, we used 2 different fluorescent proteins, and we quantified the peptides seen in single cells via SCOPE-MS, and then we also looked at their fluorescence. Now this was great. It allowed us an external verification, but there are only so many different fluorescent proteins you can quantify per single cell. You certainly can't quantify a whole proteome worth of them. So this is a great way to do external comparisons, but we want to do other ways as well. But there are other types of comparisons we can do too. We can look at internal validations of our data. We can introduce this idea of including control samples. We can also look at relative reporter ion intensity. Now that's the reporter intensity coming from these barcodes that we talked about earlier. We want to basically use this as a metric for ascertaining whether or not we're in the right range of quantitation. We can also look at the consistency of protein quantitation and use that as a metric for verifying whether or not we can rely on each individual protein's quantitative information. So I'm going to go into each of these in more detail now. So with respect to the control wells, we can include them from the very beginning in our whole sample prep process. That means that all the signal that we see in our control wells will capture any sort of potential artifacts that are introduced by the sample prep process. So we include them, we subject them to every single step in the process. They get all the same reagents. And therefore, any signal that we see in there will be indicative of the background that we might have. So I'm going to sort of concretize this now with an experiment that we did in the lab. And we're going to try and figure out, did we succeed in preparing some useful single-cell proteomic data, or did we fail, and where did we go wrong? So our experiment system here was we had some monocyte cells grown in culture, and we wanted to look at the difference between monocytes and macrophage cells that we could induce to differentiate from these monocytes. We also wanted to look at the heterogeneity in the macrophage population. So we differentiated these cultures for 72 hours with PMA differentiation, and then we sampled these 2 different cultures. We sorted them into 62 scope sets using the same plate format that was shown earlier. We randomized them, including more macrophage cells than monocyte cells because we were more interested in them. We wanted to profile more of them. And then we also included control wells in these sets so that we could look at the background signal. Now, this is sort of a good moment to stop and talk about one of the potential artifacts that can be introduced in Scope 2. Now, these commercial barcodes that we use are amazing, but they do have some carryover and contamination into adjacent channels. About 1% of all the signal coming from each barcode carries over into adjacent channel. That's not a problem when the signal in each of the channels is pretty similar to one another. But when you have something like the carrier that's way more abundant, just 1% carryover into an adjacent channel would completely swamp the signal. So therefore, we leave the adjacent barcode to the carrier channel empty. So here we have sorted 62 scope sets, and we acquired some data. It looks really good. We looked at a low-dimensional representation of all the proteins that we quantified in each of these single cells. We profiled about 350 single cells. We saw about 2,300 proteins in these. And based off those proteins, we were able to separate out the 2 different cell types. We did naive PCA separation, and then afterwards we colored by the culture that we knew we had grabbed the single cells from because we had control over that. So we're very happy. This looks like a good indication that our data is reliable, but is it really? We want to examine it with some of the ideas that we talked about earlier. So first, we're going to take a look and use those control wells. So here on the y-axis, I'm showing the relative reporter ion intensities. That's the intensity that we use to do the relative quantitation, normalized to the carrier channel. So in this case, we've normalized every channel, including the carrier itself, to the carrier signal. So that means that the carrier was then a 1 on the normal scale. Then when log 10 transferred, it became 0. And what we would expect in the log 10 scale is that when we have a carrier channel of about 100 cells, our single cell should be roughly 1/100th of the signal. That gives us a good starting point to figure out, did we have any massive contamination in our sets? Did we have no signal at all? And did we have any cross-labeling potentially from our single cell reporter ions to the carrier channels? If we get roughly a ratio around 1 to 100, what we would expect, we have more confidence that we've successfully prepared a Scope 2 set. So here we're looking at one set. We're going to look at each of the single cells in that set. And we can see that roughly we're at about 1 to 100, -2 on the log 10 scale for each of these single cells. But we also included a control well. And the control well shows much less signal, much less background. And that's really great. So that means that we didn't have any massive contamination and that we can trust that our background signal is fairly low. But it could also show us where we failed to prepare a single cell well. This failed well should have been a monocyte, but we can see that it has a highly similar profile to the control well. And we can use that to figure out if we did not successfully prepare single cells, and we then filter them out of our analysis. So another type of internal verification is looking at the consistency of quantification of the proteins. So for that, we're going to use the coefficient of variation, which is the standard deviation divided by the mean of the relative levels of each peptide belonging to each protein. So a nice consequence of doing bottom-up proteomics is that you'll often get more than one peptide mapping uniquely back to an individual protein. So for protein I here, maybe we have n different peptides that uniquely map back to it. From those n different peptides, we would expect similar relative quantification. And we can use the similarity of their relative quantification to define that as a metric of goodness of overall quantitation. So here we take the relative quantitation of peptide 1 for protein 1 in each single cell, and then we can create a distribution of CVs, each of the points in this distribution corresponding to the CV belonging to a protein from which we had more than 3 or 4 peptides. And then what we can do, and what I'm going to show on the next slide, is we can take the median of this distribution for convenience and start to use that to pick apart successfully prepared single cells and not successfully prepared single cells. So just as a recap, we're going to look at the CV for each protein in each single cell. We're going to use that to compute a distribution of CVs for each single cell. And then for each single cell, we're going to take a median value of CV and plot that on the next slide. Cool. So now we're going to look at the two slides that we just previously saw. We're going to look at the median relative reporter ion intensity for each of those single cells. And then we're also going to look on the x-axis at the coefficient of variation, which I just talked about. And what we can see is that these control wells actually allow us to start to define some heuristic values from which we can use to separate out our poorly prepared single-cell wells and our successfully prepared single-cell wells. We can see that consistently, these control wells have much higher coefficient of variation, meaning the peptide signal that we got from them doesn't agree with one another for the proteins that they should have belonged to. And then likewise, we can see that the signal coming from our successfully prepared or control wells is consistently lower than those wells that we sorted single cells into. So from this sort of metric, we can define metrics of success. And we can find that we prepared about 80% of our sorted single cells successfully. And we can use that data to have higher confidence in our later interpretation of the quantified proteomes. Great. So we've covered internal consistency, we've covered internal validation, and we have more confidence in our data now. But let's take a further step and really be more confident that we can trust our data. So here we can do external comparisons. We can compare our single cell data and compare it to bulk proteomic data. What we can do there is for each protein, we can then look at its mean value across all of the single cells of one type and then all the single cells of another type and calculate that value for each and every protein. So each and every protein across all the single cells, we're going to get an average in silico average of the protein level. And then we can compare that average ratio of this proteins profiled in one type to the relative ratio of them profiled in a different cell type and compare that to data we could get from bulk proteomics. When we do this sort of calculation, we do an in silico average of the proteins, and we compare it to bulk proteomic data that we acquired. We find very, very good agreement across many proteins. So therefore, we have much more confidence that the data that we've acquired is reflective of true signal. Cool. So at this point, we were pretty happy. We wanted to then pursue the analysis of this data because we thought that it demonstrated good quantitation. But how could we have failed? There are many points that we should talk about in the Scope 2 process that could introduce artifacts that we should consider. So just to go through them, different positions that you sort your single cells into in the high-throughput plate that you want to use could introduce biases. You could have incomplete digestion using the enzymes to break your proteins into peptides. You could have incomplete labeling, meaning attaching the barcodes to your peptides. You could have problems with chromatography related to sample carryover. Even the best chromatography is not going to be rid of the sample that you injected on it by the next run. We'd have differences in peak sampling. And then with regards to the mass spec, we could also have low sensitivity, a dirty instrument, or we could have coisolation of multiple peptides producing mixed quantitative data. And then finally, with data analysis, we could also have problems combining our different sets together if we didn't use a reference channel. Now, the reference channel helps us address the peak sampling issue, meaning sampling our peptides at slightly different points in their elution profile. And it also helps cover combining sets. So since we've discussed the reference channel already, I'm not going to talk too much about that for the remainder of the talk. But I'm going to try to address the rest of these. So first, optimizing the LCM-SMS. The first thing that we did was we wanted to make sure that our mass spec was even capable of taking single-cell measurements. So we made the ideal single-cell set, one that had no biological variability in it. We did this by creating what we're going to term a 100x master mix. And we created that using the Scope 2 design. We had two different carrier channels of two different cell types at about 100 times the amount that we want to inject. We then had single-cell channels that we created at 100 times the amount we wanted to inject. So therefore, when we take 1/100th of this sample that we created, we then have an ideal single-cell set. 50 cells in each of these carrier channels, and then 50 cells of two different types. We included two different types of cells because we wanted to be able to do and compare the relative quantitation that we could get from this. We included two different carriers because we had more trust in the measurement we would get from the carriers, more trust in the relative quantitation that we would get from these carriers, and then therefore could compare the quantitation we got in the single cells to the quantitation that we could get in the carrier channels and use that as its own internal validation. You could also use external validation, as I described before, to compare the quantitation from these single cell channels. So we ran these and we iteratively optimized our LC-MS using this 100x master mix. And Gray's going to go into a lot of detail in his workshop talk on how we use do-MS to iteratively do this process and come up with the best parameters possible for both the sample prep and the LC-MS. And then the second question is, was our LC clean enough and our mass spec clean enough to do single cell type measurements? For that, it's actually harder to assess because the carrier channel allows us such great sensitivity that just running blanks on our LC-MS is not enough to assure that we don't have spurious signal on it. Therefore, we wanted to run a carrier that was similar enough to the cells that we wanted to profile, and then leave all of the other channels empty, unused. Therefore, we'd be triggering measurement on the background signal that's remaining on our LC-MS for the peptides and proteins that we're interested in, and then we have the strong expectation that we should get no signal in the single cell channels. And here I'm showing a plot from do-MS that shows just that for most of these samples. But we can see, I show 4 replicates here of our carrier-only sample. We can see that we have high fraction of missing values in our single cell channels here in the same order as over here. And then we have some samples where we had some signal in the background, and we needed to run more blanks and clean our system better to assure that we had accurate single cell capabilities. So what were some of the issues that are beyond the LC-MS? So other issues that we talked about were introducing biases based off of plate positioning. And what we want to say about that is basically ensuring that any of the potential batch effects that could be introduced by scope 2 should be tried to be avoided by randomizing the different cells that you would want to across the different batches. So we never want our treatment, our control conditions to correspond or correlate to any potential batches. So here I'm showing sort of 2 really big faux pas where we sorted cells of one type onto one plate and cells of a totally different type onto another plate, and then process them separately, giving us 2 large batch effects that we would not be able to attribute the signal to cell type or to batch. So the solution to that is fairly straightforward, randomizing on the same plate. So another issue that we can talk about is coisolation. And coisolation happens when we sample more than one peptide at a time. Now, often this happens when we sample signal that corresponds mostly to one peptide, and then a little bit of the signal from a different peptide is included in that measurement. That's because the mass spec is not infinitely precise. And what we have, fortunately, at our disposal is a lot of different search engines are very, very good at annotating the spectra that we can then acquire and figuring out all the different possibilities of which of those fragments come from our peptide of interest, and if there are any other fragments in the spectra that could be coming from coisolation, and then quantifying the amount of coisolation that we could see. This is just one example from MaxQuant, but other search engines can do this as well. And we can assess that directly. We can look at our spectral purity, and here I'm going to plot a distribution of the median spectral purity for each of the 62 scope sets I showed earlier, and we can see that it's very high. I'm showing a scale from 92 to 100%. Most of our MS scans that we get quantitative information from are very pure and not subject to coisolation. And we have the ability to see if there is coisolation, and we can throw out those spectra that have been contaminated. Further diagnostic things we can do is we can start to address digestion efficiency and labeling efficiency by looking at the relative reporter ion information and seeing if that jives with our expectations. And we can also look at the inclusion of any small bulk samples that we included and processed in tandem with all of our scope sets. And we can look at the digestion efficiency and labeling efficiency of these small bulk samples that we included and processed in tandem. So in summary, we talked about the 2 sort of design principles that we want to keep in mind when designing scope 2 sets. We want to figure out if we succeeded, if we can trust our data, and if not, how can we include lots of controls to figure out where we failed. At this point, I'd be happy to talk with anyone in person about any of these metrics that I've confused you on and take any questions. Thank you.
Okay, I see there are quite a few questions. I'm going to ask Sebastian to help me with the mic.
So in the slide when you were showing how to use the control wells to to basically, yeah, exactly. So it seems that there's a range of the relative ion intensities. Where does that variation come from for the reporter ions?
So you're talking about the whole distribution that we see here.
Correct, yeah.
So one source is biological variation. And as you can see, we can compare it to the control well and see what sort of expected variability that we would get from artifacts in our data. So if you're talking about these peptides up here, that quantitative information could come from differences in signal between the carrier channel and the single cells themselves. Or it could mean, for example, we could have a protein that's truly spuriously high in our single cell. Or it could come from contamination signal, because we can see that we do get a very small amount of signal that does belong to that whole spread of range that is introduced as an artifact in our control well.
So this is the reporter ion intensity across all the peptides of a sample?
Yes.
Okay.
Other questions?
Why don't you directly use peptides as a reference channel?
You mean synthetic peptides, or?
Yeah.
We certainly could. But then if we wanted to basically use the reference channel to normalize the whole proteome, we'd have to have a whole proteome's worth of synthetic peptides.
But if you're familiar with DIA, data independent acquisition, you will know that we tend to use something called IRT. It's a series of human and synthetic peptides, and it's very precise, even able for RT normalization.
Sorry, what's the question?
Retention time normalization and intensity correlation. If you're using IRT, the DIA stuff.
No, so the type of acquisition that we do is data-dependent acquisition.
Can I just follow up? Kudos to both of you for putting up your preprint, which I will read closely. But until then, so the separation in signal between the empty wells and the single cell wells looks like maybe a log or an order of magnitude in the mode. That's correct?
So basically, a lot of the signal that you see down at this -3 bin could correspond to the cutoff of the search engine for the signal-to-noise ratio that it's willing to supply you with as it's done the peptide quantitation.
No, I just mean if you look at the actual spectra in the reporter range, and you look at the intensity of those reporter ions. If I'm interpreting this y-axis correctly, it looks like the difference in the reporter ion intensity is about 10-fold between the empty wells and the true single cell wells. Is that correct?
It would appear that way, but what I'm saying is that this -3 bin actually may contain a lot of missing data.
Let me ask you, the question I'm asking is, what's the overlap in the area under the curve for the distribution of reporter ions in the true single wells and the apparent reporter ions in the empty wells? Yeah? You see what I'm asking?
Not quite.
Okay, maybe I'll come and ask you after.
Yeah,
so
I'd be very
if you were to plot
interested.
the distribution of reporter ion intensities, it follows some distribution.
Yep.
So I think I have a version.
Yes, thank you.
So in the control well, over 95% of the reporter ions are going to have signal-to-noise ratio of essentially 0. So the signal that we measure in the control wells is equal just to the noise threshold of the instrument.
I agree. What's that separation between that signal, which is there? It's electronic noise, it's ambient ions, and the true signal from the single cell.
So I think...
So if this is the false distribution, or the background noise distribution. This is the single cell distribution. What's the...
Nikolai, do you want to talk about the ion counting plot?
So your question, maybe another way of phrasing it is, what is the signal-to-noise ratio for reporter ions coming from the signal, from single cells? And on average, we count over 100 ions coming from the single cells. which is about 2 orders of magnitude higher than for single-cell RNA sequencing. So signal-to-noise ratio will correspond to about 100 ions coming from the single cells on average for most peptides, or more. And that is for a particular, I'm sorry, maybe going here a bit on a tangent, but this is for a specific instrument settings that aim to optimize both quantitation and throughput. If one wanted to accumulate more ions so that you have higher signal-to-noise ratio, you could simply accumulate them for longer, and that would result in having higher signal-to-noise ratio at the expense of being able to analyze fewer peptides per unit time.
Really great talk. There was one slide, and you kind of went past it pretty fast, but on there it said that you quantified 2,300 and some proteins. Is that how many proteins you saw across all samples? Was that the average that was seen within? Which one is it?
Yeah, so take a look at the preprint. It answers all those questions, I think. We will give you the average number of proteins seen in each single cell, and then we'll look at also the number seen across all the single cells. 201 protein, or 21 proteins is the amount of data that we were comfortable imputing to obtain a complete data matrix with no missing values. So that's not a straightforward answer.
What was your threshold for imputation? What percentage of samples did it need to be seen in?
Let me think about that. We had a fairly high threshold for imputation. It was roughly 95%. We allowed up to 95% missing data.
Up to 95% missing data, so it only needed to be seen in every 20 samples for imputation.
So
Okay.
what we had was 350 single cells, and we did k-nearest neighbors imputation. And what we needed was hopefully just a few nearest neighbors in order to get reliable imputation. Now, 1 in 20 of 350 gives us more than the number of Ks that we were considering, and more than certainly the number of cell types that we were considering. But we can do this for any number of parameters and tweaking the data analysis. So I'd be curious to talk about what would be a comfortable range, and we can look at how the data looks under those parameters too.
Great, thank you.
Thank you, great talk. Could you please show me the PIF figure?
Which one?
PIF impurity.
Yep.
I guess it's in the last part of your talk. Could you please tell me what is the exactly column you used, pharmafluidics or other columns, and what is the gradient length? Because I didn't see this even in bulk sample experiments in my hand.
Yeah, so the column that we used to do this was the Ion Optics Aurora series column. We've been super happy with the peak shape that we can get from that column. It is incredibly, incredibly sharp. It's better than so many different columns that we tested over time. It's truly amazing and definitely responsible for the high purity of spectra that we can get.
Yes, so you say the Ion Optics, I guess 25 centimeters?
I think we're using the 25 centimeter version. They have 2 versions. I can double check with which length we used, and then we use a 60-minute LCMS, about a 45-minute active gradient.
Another question, what is the ion accumulation time after you use the DOMS platform?
Yep, so we allow accumulation times up to 300 milliseconds for MS2.
300 milliseconds. Okay, thank you.
Hi. How did you actually define these particular numbers from MS1, I assume, of these 60-minute runs?
Yeah.
Based on whatever that max quanta search engine just gives you as a number, correct?
Yep.
Yeah, and then in this particular experiment, you have 100 cells as a carrier channel. Yeah. That's basically the question is here. Imagine instead of 100 cells, you will have 1,000 cells, 10,000 cells, or million cells. Try to run that on 60-minute gradient. You will see your impurities.
Yeah. So I think what Bogdan's talking about is that the complexity of the proteome is basically going to affect this measurement and our ability to have pure spectra as well and measure the purity of our spectra. The deeper you go in the proteome, the more proteins you're going to uncover. So if you inject an incredibly intense sample on a very short gradient, you're going to see many, many peptides per unit time, rather than we inject a less intense sample, and we're only profiling the most abundant proteins. So therefore, our spectral purity is higher, or our ability to measure the spectral purity results in these sort of numbers. Oh, yeah. Yeah, and we have achieved much lower spectral purity on these same type of samples. Using the master mix, we spent probably at least a year optimizing our LC-MS parameters, and that whole year resulted in the creation of the software that Gray is going to talk about for doing that process.
Very nice stuff. So, of course, now you just launched Scope 2, so I'm sorry to ask this question, but like, what is the future? I mean, in terms of design of the method, are you trying to go for, let's say, losing the carrier, or at least minimizing the need for the carrier, or minimizing the number of reference cells? I mean, there's many applications where we cannot or do not have cells that are somehow similar, right? I mean, in fact, what we want to look at is the differences. So I'm just wondering where you're going with this in sort of future design of a method.
Yeah, so there's lots of flexibility with the carrier design, and you can induce measurement on whatever proteins you'd like to put in it. And we have the ability to add synthetic proteins, synthetic peptides to the carrier channel. So there's lots of flexibility in designing it. I think that if we had unlimited barcodes, we wouldn't need a carrier channel. We would simply have over 200 single cells profiled at once, and that would be the exact same amount of material that we're profiling now. So that's one direction that we could go. The other direction is designing different carriers. I hope that answers that question.
Yes, thank you. I have two questions. The first is a quick question. What's the lowest abundant protein you identified ever?
The longest?
The lowest abundant protein you ever see by single-cell proteomics.
So we can quantify the copy number of ions that we're able to profile, but as far as assessing which protein we expect to actually have the lowest abundance in the proteome, that's a harder question to answer because peptides have different ionization efficiencies. So the number of ions that we can actually get into our MSMS is not proportional to the number of peptides that are in the proteomes of interest. And that's why a lot of our analysis is reduced to relative quantitation, because of course they're all ionized the same and the relative levels will be similar, but we can't sort of answer that question directly.
Okay, the second question is, how do you convince people to use single-cell proteomics over single-cell RNA-seq?
I mean, if you're interested in proteins, you're interested in proteins. I think that I'm not a single-cell RNA-seq expert, and I think there are just different types of experiments you'd want to do.
Hey, Harrison, I was wondering if you could go back to that plot with the reporter ion and the order of the labels that you have in there.
Yeah.
That's
this one?
No, the one with the distribution. Yep, that one. So can you comment about what types of experiments and evaluations you did in order to choose that the carrier reference or the carrier will be in the first channel, and where you place the blanks in the controls, and how you decided upon those and why those might be the best way versus scrambling the order of the reporter ions and the labels?
Yeah, great question. So we fixed the carrier channel with one label because of the carryover into an adjacent label. So we wanted that to sort of be on the end. We wanted that to be in 126C. So that's the choice of our carrier location. The reference location is likewise fixed because since it's slightly more abundant, we still don't want any carryover from that as well. And so that fixed that channel. The rest of them we did randomize. So we randomized the control well and all the single cell wells before we sorted them, and then implemented that randomization scheme when we sorted the cells. So the rest of it has been randomized. Yeah. And then as far as, I think you asked about the carrier channel amount. We did spend some time investigating this, and we reached a happy point for our instrument and our experimental setup. But I think that that is something that can be investigated, and certainly may be different for different cell types.
Hi. Can you comment on how long it takes to do the wet part and the computing part?
Yeah, so the actual, let's go back to sort of the overview here. So you have cell culture, which takes as much time as it needs. The sorting itself and this whole process, we detail in the bioarchive. And the sample prep takes about 1 day, about 8 hours. Sorting itself, if it goes well, can be very quick, or it can be onerous. The process right here itself takes about 6 hours. And then the mass spec analysis is 90 minutes per scope MS set. So that resulted for these 62 sets, we had about 85 hours of LCM SMS time to get 350 single cells.
Hi, great talk. Just a quick question, tagging along the sample prep. So how do you process basically the breaking down the cells from step 1? And as I understand, you'd use an acoustic sonicator. Does that method to use for breaking down the single cells, would that change when you use carriers because there are more cells, so you use a different pulsing methods, or they're all kind of same all across?
Yeah. Great question. So when we did Scope 2, Scope MS uses this sort of high energy focused acoustic sonication. And that method is pretty low throughput, and it requires a very expensive instrument. So for Scope 2, one of our real big motivations was to make it cheaper and more easy to do for everyone. So we do the lysis with this process called MPOP, which is just a freeze heat lysis that we validated in the very same wells that you sort the single cells. So it's very easy. You can sort your single cells into whatever well plate you want. You subject it to a freeze heat cycle. The heating is pretty high. And then that is sufficient, it turns out, to extract proteins from our single cells, especially when we then digest them.
What is the extraction efficiency of your buffer?
Yeah, so we have another preprint on bioRxiv where we go into this. We called it MPOP for minimal proteomics preparation. And the extraction efficiency across most of the proteome is comparable or better than another different extraction efficiency that you could achieve with, we compared it to urea. So we directly compared the extraction efficiency across the whole proteome to an extraction with urea-based lysis buffer. And we found that MPOP was very comparable.
How do you find that MPOP performs for, say, membrane proteins as compared to cytoplasmic fraction? The cytoplasmic fraction is a whole lot easier to get to than a membrane.
Yeah, so we wanted to figure that out too. And it turns out that a lot of the regions that are inside the membrane we're not going to analyze by mass spec. They're not amenable for tryptic digestion, and they don't fly well on the mass spec. They have long ranges of, I think, lysine. So we are able to see proteins from that dangle on either side of the membrane, though. We're parts of the protein and peptides that come from parts of the protein that are on either side of the membrane. So we are able to see those, and we, to a limited extent, analyzed the comparison between MPOP and urea related to membrane proteins and found that MPOP performed comparable or better in most cases.
Great, thank you.
Have you seen the ratio compression effect in the single cell data?
Yeah, so ratio compression might happen when we are comparing things that are in different quantitative regions. So if you can imagine, sort of, we can have a hockey stick when we're looking at the relative amounts of signal that come from our molecules. So we might have one region of our measurement that is fairly flat or fairly shallow curve, and then we might have another region that is much steeper. And the ratio compression happens when we compare points on those two different parts of the curve that are very far apart. But when we're fairly close to one another in relative levels, the ratio compression is much less. So I think we have the potential to experience ratio compression when using the carrier channel as the reference, or using the carrier channel as the denominator for comparing different sets. But when we use the reference channel, which is much closer, and we're much more guaranteed to have it be in the same linear range as our single cell quantitative information, we have a much higher guarantee that we're not going to experience ratio compression, not to at least the same extent.
Hello. I have a question about the cell sample preparation. So when you're talking about the single cells, it's coming from the culture cells or from tissue sample prep? Can you do tissue, so frozen or fresh?
This is a great point. So we've validated MPOP, this sort of lysis and extraction procedure for cells that are in suspension. And we think that it'll work for any cells that you can get in suspension well. So that doesn't mean that it'll be directly applicable to tissues in cells that aren't in suspension. So that's the experiment that we did. We used monocyte and macrophages. Monocytes grow in suspension. Macrophages are adherent cells, but we then got them into suspension before sorting and then processed them with MPOP while they were in suspension.
Hi. So quick question first. The spectral purity you mentioned, is that one of the many columns that comes out of the max quant PSM table?
It comes out in the evidence table.
Okay, great. And then I would just like to make a quick comment about data imputation. It's very natural to want to avoid data imputation at all, and I would just warn that if you're right at the edge of the limit of quantification and then you do simply ignore missing values, you can unintentionally overestimate the average quantity because you're only averaging across the quantified values and you're ignoring the ones that might have been truly low or below the limit of quantification.
That's a really good point. And yeah, the shape of the missing value distribution for proteomics is really interesting too. But I just want to point out that this in silico averaging that we did and then comparison, this was on data that didn't use any imputation. So we ignored all the missing values. That doesn't answer any question that you asked, but it's just related.
Yeah, I would like to have a couple of comments for different questions because I finally got a word. First of all, MPOP was validated for the tissues. It does work. It's been done in our group, and I will present it tomorrow. That's why MPOP working for the tissues. Number two, missing points. We have to be careful because mass-spec is a trap. Not only for the ions, but for everyone, traps, because it's a very, very complicated statistics. It's very hard to do imputation without introducing the false positives. That's why it's very, very hard to figure out what is the threshold when you can or where you cannot do the imputation. Number three, by using the median of the cell population, you do lower your biological signal, because unfortunately your cells are not fixed. They are sitting in a different phase of the biology, right? And different phase will have a different type of protein abundance, which you average out by doing median. That's why you have to be very, very careful to do mathematical statistics applies to the real biology, just as a comment.
Yeah, so I think that brings up a really good point, though, that we don't have to do imputation at all if we don't want to. So Nikolai is going to show a plot later that shows the number of proteins that we can identify in at least X cells. If we want to see the proteins that are seen in at least 5 cells, we can actually see a lot of different proteins. And we can just limit our analysis to those cells that we've truly observed it in. So we're not subject to doing imputation in order to get usable and interesting biological information out of our single-cell proteomics data. We simply have to measure it. Now, we do imputation because imputation, we are required to have a complete data matrix in order to do PCA, and everyone likes showing PCA.
Yeah, this will be a question and a comment. As mass spectrometrists, as many of us in this room, or at least some of us, handful, all right, maybe there's only 3 of us, in this room do, we often get ourselves into a box, especially with imputation, where we start saying, oh, the statistics of what we do are very difficult. What I'd invite everyone to do is to look at the raw numbers from an RNA-seq single-cell experiment. RNA-seq single-cell is exploding right now. Everybody wants to do it. And so let's think about how many things they're counting. They're counting, on average, there's what, 15 mRNA in a cell? So they've got huge counting error that's already included in that. If you look at the raw matrix, 99% is empty in a single-cell RNA-seq. And yet they come up with beautiful, beautiful distributions in PCAs by applying really, really interesting and advanced algorithmic techniques. And so we shouldn't be shunning that because we think those methods are impure. We should be embracing those supervised methods because ultimately, this is infancy. This is an enchote technology, and it's already doing incredible stuff. We're already at the depth that RNA-seq can see out of T cells at 2,300 proteins. That's all it can see. And they're never going to be able to cross that threshold, whereas we can. So I invite everyone who is here at this conference, please think outside the box because there are a lot of health issues that we could apply these to really help people. All right, and think about it that way. Thanks so much.
Yeah, and the rate of engine on that too is that the rate of missing data that we have is also dependent on experimental parameters, LCMS parameters, that can also be optimized to obtain less missing data. Here we wanted to use a method that gave us high throughput, but we could also basically devote much more LCMS time per our proteins of interest, target them, and acquire much more of the ions. We only sample a very small portion of them to get these measurements. We can sample much more of the available ions that we already have and acquire much lower missing data rates. In fact, probably close to complete data. Cool.
I had a quick question about your reference channel. You said that you had to prepare a sample for a reference for each cell type that was represented. If you were working with a much more heterogeneous sample like a tissue, would you need to have a reference for each one, each cell type that's represented in the tissue? Or I guess a different way of asking the question is, what would happen to your reference if you use the same reference for both of the cell types that you had represented?
It's a good question. So ideally, when you design the reference, you want to design it in such a way that it contains a protein amount for every protein that you're interested in. That exact amount just needs to be enough to be quantified. If we can quantify it, then we can use it as a consistent denominator across all of our different cell types. But that's a great question. We may have cells that have different sets of proteins in them. And in that case, we would want to make our reference as a mix of those cells. And if that's not possible, we can even then add in proteins as well. But that's an important point, that the proteins need to be in the reference in order for us to get quantitative information from them.
I would just have a quick question. Don't you think you will have a quite imbalanced ratio compression when you have on one side of your labels, your carrier, your reference, and your empty channel throughout bleeding over from the carrier? So you will have quite abundant signal on the one side and quite low abundant on the other side. Don't you think that introduces more noise?
So are you talking about bleed over from the carrier into the reference?
Exactly. No, in the unused one.
In the unused one.
You will still have a lot of signals throughout isobaric impurity, right?
Yes.
So you will have quite abundant signal on the first three references, and then quite low signal. Don't you think that introduces quite imbalanced ratio compression throughout the reported channels?
It would if we were using the carrier channel as a denominator for quantifying the single cells, I think.
But you use the reference one, right?
But we use the reference, yeah.
And don't you think the reference will be compressed by being flanked by the carrier and the unused one?
No, I think that they're sufficiently far apart in the M/Z range that the peaks, the signals will not merge from those. I think I know what you're asking, but I'm not entirely sure. So you can get ratio compression when you have peaks merging in the M/Z space. Is that what you're talking about?
partially, but
Okay.
also the compression as cannot be resolved by the mass spec.
Yes.
These 100 to whatever 5.
Yeah,
ratio.
so basically the mass spec's not taking perfect measurements at each m over z. It's actually a Gaussian distribution, and when you have them very close to one another, those then merge, and then the peak separating algorithm can't make heads or tails of it. But we have our carrier in 126 way over here. We have the bleed over from it, which is about 1%, so about a single cell level, into the 127 pair, which are much closer together. 126 is way over here. The two 127 barcodes are much closer to one another, but separated from 126 and 128 by a good margin. A greater margin then could be gapped by poor resolution on the mass spec. And the amount of signal in the reference and the bleed over is very comparable.
That's great. I love it. So many questions. I really think we are off to a great start.